We are going to begin the second session, and I want to welcome our next session chair, Dr. Don Diamond. Dr. Diamond is a Ph.D. or is a professor in the Department of Hematology and Hematopoietic cell transplantation. His research includes developing vaccine to combat hematologic malignancies, solid tumors, and infectious pathogens such as herpes virus, cytomegalovirus, and HIV. He earned his PhD from Harvard Medical School and Mass General Hospital. He has 38 US patents and over 170 original papers. He has served as a faculty member and a scientist for over 30 years at City of Hope. Welcome, Dr. Damon.
So this morning, we're going to continue with three excellent investigators. And I do have a story from the middle one, but we'll wait. So the first speaker is Dr. Münz from, I believe, Switzerland still. I have two cataracts, so I have to put this a little bit closer. In a couple of weeks, I won't have them anymore, but now I still have them. So he's going to be talking about control of oncogenic human Gamma-Herpesviruses, something, of course, those of us in the herpes field are very interested in. And he is joining us from Die Schweiz, or Switzerland, and he is a full professor in the Institute of Experimental Immunology, medical and science faculties at the University of Zurich in Switzerland. And he's a faculty member since 2008. He's also been faculty at Rockefeller, and he did get his PhD at a very distinguished and a little bit older university than some of ours in the States, the University of Tübingen. He's, of course, won numerous awards, including the Sobek Award for MS research, and interestingly enough, a Young Investigator Award from the Arnold and Mabel Beckman Foundation. He's been co-chair of the annual Congress of the Swiss Society for Allergy and Immunology, and president of the Swiss Society for Allergy and Immunology. He is well published, over 300 articles and book chapters, and he has a very enviable H factor of about 80, as I recall, with over 43,000 citations. And he's younger than me. So, without further ado, Dr. Münz.
Thank you very much for the invitation and the nice introduction to your very interesting symposium. So I would like to talk about my favorite research interest, which is indeed the immune control of human Gamma-Herpesviruses. So obviously, many of us that are interested in this field are puzzled by the fact that these two human Gamma-Herpesviruses, Epstein-Barr virus and Kaposi-sarcoma-associated herpes virus, are very widespread in certain populations. CBV, more than 90% of the adult population carries the virus persistently, and for KSHV, at least in sub-Saharan Africa, a large amount of the population with seropositivity rates of 80% carry KSHV. However, fortunately, only very few individuals develop pathologies that are caused by these viruses. And one of the main reasons for that is that our immune system is quite capable of controlling these viruses in most infected individuals for life, for very long time periods. We would like to understand how this immune control is achieved, because it seems like that it's desirable to mimic this immune control against other disease agents or maybe also cancers in general. However, nevertheless, Epstein-Barr virus, for example, causes a wide variety of diseases, as mentioned in a previous talk, in between 200 and 300,000 new tumor cases every year that are associated with Epstein-Barr virus. And some of the diseases originate from the main host cell of this virus, the human B cell, such as Burkitt's lymphoma and Hodgkin's lymphoma. However, there's a second class of pathologies that emerge from other cell types, either epithelial cells, as in the case of nasopharyngeal carcinoma, or other lymphocyte populations like NK and T cell lymphomas. We have a much less developed understanding how these tumors are actually caused by Epstein-Barr virus. Then there's a third group of pathologies, which are immune pathologies, so overshooting immune responses to EBV infection. And the most common one is primary asymptomatic EBV infection or infectious mononucleosis. But these immune pathologies can probably also extend into autoimmunity, and the autoimmune diseases that is most frequently discussed in this respect is multiple sclerosis. You might be aware of publications last year that suggested that when you follow in the American military personnel cohort, and this is a study by Alberto Ascherio's group, and you look from the clinical onset of multiple sclerosis in these patients back in time by samples that have been collected prior to clinical onset of MS, that you see uniformly EBV infection on average 7 to 8 years prior to the clinical onset. And in these individuals that will develop MS, then in these prodromal phases, so in the preclinical phase, one can see after EBV infection already signs of neuroaxonal damage. So quite curious, one virus, right, that seems to cause a multitude of diseases, and we would like to understand how we can counteract them and how we can actually reestablish immune control that works so well in most of us that carries the virus asymptomatically. So Epstein-Barr virus is a human gamma-herpes virus that is thought to be transmitted primarily via saliva exchange and finds then in submucosal secondary lymphoid tissues, such as the tonsils, its main host cell, the human B cell, then basically develops two strategies for its own amplification. One is through so-called latent infection. It drives its own amplification through the proliferation of B cells. B cells that are driven into this proliferation through either a combination of nuclear antigens, so-called EPNAs, and latent membrane proteins. So there are 6 nuclear antigens and 2 latent membrane proteins. And the goal of this exercise seems to be that the virus can access the memory B cell pool through B cell activation and differentiation. And in this memory B cell pool, then the virus persists. And in most healthy virus carriers, around 1 in 10 to the 4, 1 in 10 to the 6 B cells in the peripheral blood are infected with the virus and carry it in the bloodstream. Once from this reservoir, B cells get activated into plasma cell differentiation, so the terminal differentiation for antibody production. Then that switches on lytic replication of the virus. The virus then is thought to maybe undergo a second round of lytic replication in the epithelium of the mucosa for efficient spreading and transmission. The slide also shows that all the malignancy states or the gene expression programs that are found in different EBV-associated lymphomas, such as Hodgkin's lymphoma and Burkitt lymphoma, are already present in healthy EBV carriers. And therefore, it is thought that the transition from these pre-malignant states into overt malignancy is blocked by our immune system. And at least for EBV, because it's so widespread, we have a quite good idea what components of this immune control are necessary to control EBV infection in an asymptomatic carrier state. Because individuals with primary immune deficiencies or mutations in individual genes that are susceptible for EBV-associated pathologies, they guide us into a picture like that cytotoxic lymphocytes are at the core of EBV-specific immune control. The genes that are mutated and predisposed for EBV disease are, for example, genes that are involved in the development of these cytotoxic lymphocytes, so CD8 T cells primarily, but also some innate lymphocyte populations like NK cells. There are genes which are required for their expansion and survival during expansion. And then there are genes that are involved in TCR signaling of cytotoxic CD8 T cells, genes that are involved in the co-stimulation of these cytotoxic CD8 T cells and the cytotoxic machinery itself, perforin granules that are degranulated and therefore kill EBV-infected cells. What is quite curious about this picture is that for EBV-specific immune control, interferons, both type 1 and type 2 interferons, don't seem to play a role, and also antibodies are not required for EBV-specific immune control. So a control that is basically mainly based on cytotoxic lymphocytes. So what modifies this immune control that works so well in most of us in settings of EBV pathology? And some of these settings are immune compromising settings, either, for example, HIV co-infection that compromises our cell-mediated immune control, iatrogenic immune suppression, for example, after transplantation, when we apply directly immune suppressive drugs. I already mentioned mutations or congenital immunodeficiencies. There's probably also immune modulation by Plasmodium falciparum infections or in holoendemic malaria regions that is causative or associated with the development of Burkitt's lymphoma caused by EBV. Another co-infection that is involved in EBV-associated pathology is a co-infection with Kaposi sarcoma-associated herpes virus or human herpes virus 8, close cousin of EBV. In primary fusion lymphoma, a tumor that is associated mainly with Kaposi sarcoma-associated herpes virus to 100%, 90% of the cases also Epstein-Barr virus is harbored in the very same tumor cell. In addition to Kaposi sarcoma and multicentric Castleman's disease, which are also caused by Kaposi sarcoma-associated herpes virus. I want to talk, or I want to use this opportunity of my talk to talk primarily about this particular tumorigenesis, primary fusion lymphoma, and how we try to mimic it in experimental animal models, and also what we learn about the T-cell response that emerges then in these experimental settings, and what we can learn maybe about protective T-cell responses against KSHV. So the model that we are using is quite complicated because, as you know, Epstein-Barr virus and Kaposi sarcoma-associated herpes virus are exclusively human trophic pathogens. For that reason, we use mice that lack their own lymphocyte compartments due to genetic mutations. And then we neonatally transfer into them hematopoietic progenitor cells. We allow then human immune system components to grow up for several months. And in these studies where we look at KSHV infection and EBV infection together, we infect them either with KSHV alone or Epstein-Barr virus as a wild-type form of the virus that can undergo both this B-cell proliferating latent infection or lytic replication, so the lytic replication for virion production, or a virus which is actually deficient in the lytic replication because it lacks the immediate early transactivator of lytic replication, BCLF1. We compare usually 4 groups, uninfected. We tried KSHV only infection, which, as you will see, doesn't work very well. EBV single infection and co-infection between KSHV and Epstein-Barr virus. When you look in the co-infected mice, you can, for example, identify KSHV-infected cells by LANA staining, so a nuclear antigen of the KSHV virus, the DNA maintenance protein. You can see that in these mice, there are CD20 positive B cells that are infected, but also IIF positive or plasma cell differentiated B cells that are infected, and that will become a little bit important later on. To our surprise, when we started these studies, we found that KSHV infection alone is only perceivable after months of infection in a minority of mice. So persistence does not seem to establish very well in the single infection. And when you look actually in these KSHV single infected mice, and also in the majority of cases or in the few mice that can actually be still detectable for KSHV viral loads, the viral loads are very low. However, when we co-infect with Epstein-Barr virus, either with a wild type or the virus that lacks lytic replication, then we find that the majority of mice now can actually sustain KSHV infection, and the viral loads come up to quite detectable levels. When we now look where this infection takes place, and I would like you to focus initially here on this double staining for the nuclear antigen of KSHV, LANA, and the latent membrane protein 1 of EBV. You can see that in the double infected animals, primarily KSHV expression or infection is detectable in cells that are at the same time also infected by the Epstein-Barr virus. When we do that analysis more quantitatively, where we use KSHV, which drives a GFP reporter, and we look at the GFP positive and KSHV infected cells, and then stain by flow fish, so hybridization for non-translated RNAs of the Epstein-Barr virus, the EBERs, we see that the majority of KSHV infected cells are actually also EBER positive. It seems like not only individual cells in histology, but also more general in flow cytometry, we can identify KSHV infection primarily in EBV infected cells. You can also grow out these cell lines then from the in vivo infected animals, either from EBV single infected animals or from EBV KSHV double infected animals. And these cell lines that we derive from the double infected animals are able to sustain KSHV infection at viral loads that are actually not too dissimilar from the viral loads in primary fusion lymphoma cell lines that have been derived from patients, most of which are also double infected for Epstein-Barr virus. We then looked more in the gene transcription pattern of these cell lines that we derived from this double infection. And the Dalla Favera lab at Columbia had published a core transcriptional pattern or sequence that from primary fusion lymphomas samples from patients, which are characteristic for primary fusion lymphoma and distinguish it from other lymphoma subtypes. You can see here when we align our RNA sequencing of the double infected cell lines and the EBV single infected cell line with this core transcriptional pattern, then we see that indeed in the double infected cell line, those genes that are characteristic for primary fusion lymphoma are upregulated, and those that are characteristically downregulated are also downregulated. One of the main hallmarks of primary fusion lymphoma is this further differentiation in the B cell development into a plasmablastic lymphoma type with characteristics expressions of IL-4, BLIMP1, or also aquaporin. But also when we look more broadly at the gene transcription pattern, then we see that the double infected cell line clustered together with the primary fusion lymphoma cells from patients, while the single infected ones cluster with lymphoblastoid cell lines that can be derived by EBV infection in vitro. You see that they are still clustered a little bit away, so they are not exactly the same as these long-term established primary fusion lymphoma cell lines. We don't know if that is, the reason for that is either that these cell lines have drifted, or presumably also these double infected cell lines are still a precursor state of primary fusion lymphoma, and on longer duration might actually acquire additional changes. We also see that in this double infection that we develop more lymphomas in these humanized mice. Now around 50% or slightly more than 50% of these mice develop tumors, while in the EBV single infection, usually 20 to 30% of mice develop tumors. When we eliminate the lytic cycle of EBV infection by using the BCL11 knockout virus, and we see that this tumorigenesis is reduced to the tumorigenesis or the frequency that we see actually in single infection, suggesting that KSHV co-infected cells benefit in their tumorigenesis from the EBV lytic cycle, and the EBV lytic cycle conditions the microenvironment, as we have shown in other studies, presumably the microenvironment for the tumorigenesis. So how is then the immune system reacting to this double infection of KSHV and EBV, which is primarily in double infected B cells? We were particularly also interested in the T cell mediated immune control for KSHV, because even though there are much less primary immune deficiencies described that predispose for KSHV pathologies, from those one can already glance that most likely the T cell mediated immune control against KSHV infected cells might be different from the one that is actually required for controlling Epstein-Barr virus. For example, KS, so Kaposi sarcoma, was observed in deficiencies of the type 2 interferon system, which is not actually required for EBV-specific immune control. And also other co-stimulatory molecules have been identified to predispose for KSHV pathologies. So therefore, we analyzed the T cells that react to KSHV co-infection with Epstein-Barr virus. You can see, for example, here again, LANA staining, identifying the KSHV positive cells, which are these large, more plasmablastic cells, often in close proximity with certain CD3 positive T cell subsets in histology sections here from the spleen of double infected mice. When we then look at cytotoxic CD8 T cell expansion and helper CD4 T cell expansion, and we compare always single infected, wild type EBV infected, or BCL11 knockout EBV infected mice, the double infected mice, then we see that CD8 T cell expansion, both in blood but also in the spleen, is enhanced upon the double infection with KSHV. This increase is much less pronounced for the CD4 T cells. When we look at the differentiation of these T cell compartments, and in these pie charts, we have sort of CD4 cells on this side and CD8 cells on that side. Here is basically a mock infection at baseline and in blood at week 4 and in spleen at week 4. Compare that with the single infections and the double infections. And we see that the double infections drive quite massively, for example, down here, the expansion of both effector memory CD8 T cells and central memory CD8 T cells, that to a larger extent than actually the single infections. You also see antibody responses occurring. So we see an increase in IgM responses in the serum, elevated levels of IgM responses, even though these are not dramatically different between the double infection and the single infection with Epstein-Barr virus. Achilles heel of our animal model is that there's very little IgG development, so class-switched antibody are very difficult to observe, and you see that only a subset of mice presumably reacts with IgG responses to the double infection. Collaboration with Dennis Whitby from the Frederick Cancer Center, then we analyzed the specificities in the immunoglobulin responses and the IgM responses, and Dennis has expressed a large variety of KSHV open reading frames as recombinant proteins for a multiplex assay to look at antibody responses. When you look at the serum of the double infected mice, these are double infected with the BCL11 knockout virus, Epstein-Barr virus, or the wild type Epstein-Barr virus, compared to single infected animals or mock infected animals. And you can observe that a variety of IgM responses can be detected in response to the double infection. These include actually reactivities against the LANA, so OFF73 and K8.1, which are the two most widely used proteins to detect seropositivity for KSHV, but also against a variety of other antigens. However, this most likely only reflects that the B cell compartment reacts to the presence of these proteins of KSHV in the double infection, suggesting that indeed also at the protein level, the co-infection with Epstein-Barr virus leads to more antigen expression of KSHV due to persistence of KSHV. We then isolated T cells that we aim to look at T-cell specificities against KSHV. For that purpose, we use splenocytes of double-infected mice, co-culture them with autologous B-cell lines that are double-infected with KSHV and EBV. And directing T cells from the splenocytes we capture by their interferon gamma production, then derive KSHV or cell lines that are preferentially recognizing the double-infected cell line, so the KSHV EBV double-infected cell line over the EBV single-infected cell line. You can see here an ELISpot assay for interferon gamma production of one of these cell lines. You can see that it has a low level of reactivity against the EBV-infected only cell line, but then a higher reactivity against the double-infected cell line. You then clone these cell lines to derive CD4 T cell clones and CD8 T cell clones that are then now reacting with cytokine production, but also with degranulation as a surrogate marker for cytotoxicity, primarily to the double-infected cells, so KSHV, EBV double-infected B cell lines, and very little background reactivity against the EBV single-infected B cell lines. We also observe that the KSHV double-infected cell lines are killed by CD8 T cell clones that you derive that are specific for double-infected cells. We have particularly looked at this T cell clone 1-1, which also you can see here in an E to T target ratio titration kills preferentially the KSHV EBV double-infected cell line. This clone we analyzed then further again with Dennis Whitby for its specificity. Denise characterized or has overlapping peptide libraries from a number of KSHV open reading frame proteins. We observed in originally the paternal or the original cell line that we derived from a double-infected mouse, primarily K6 reactivity, a lytic antigen of KSHV. And also the clones that we derived, recognized by interferon gamma secretion, EBV-infected B cell lines only when we pulse it with a K6 specific peptides. This T cell line was then also, CD8 T cell line was then also used to look at the killing of KSHV and EBV-infected B cells in an in vivo killing assay. We inject both single infected and double infected cells that we have differently labeled with different concentrations of a fluorochrome into mice. Then we co-transfer T cell clones and after 5 hours extract then the cells again and see that basically then the CD8 T cell clone can preferentially kill the KSHV double infected cells and to a much lower extent the single EBV infected cells. And also when we deplete T cells during KSHV and EBV double infection, then we observe that this leads to an increased viral load, KSHV viral load upon T cell depletion, suggesting that also overall the T cells that we see expanding in the double infection mediated some degree of immune control. Immune control not perfect, so it only increases the viral load by 5 to 10-fold. So let me sum this up. EBV facilitates KSHV persistence in our experimental model. And this has now also been identified in a variety of other settings. For example, the Sackton's lab has shown that the double infection indeed allows persistence of KSHV also upon double infection in vitro, even though there KSHV persists only in a very small subset of double infected cells, around 1 to 2%. The Dittmer's lab has also demonstrated that in the double-infected primary fusion lymphoma cell lines, when you eliminate EBV from those, then actually KSHV DNA is also lost, suggesting that KSHV, even in these established cell lines, requires some function of EBV for persistence. And there have been epidemiological studies in Uganda and Cameroon that suggests that indeed EBV seropositivity or EBV infection is a main environmental risk factor for KSHV seropositivity, suggesting in these geographical regions that maybe even both viruses are very early on vertically transmitted from mother to child at a very, very early age. KSHG persists in EBV-infected B cells that drives into plasma cell differentiation, and these cells that we can grow out but also emerge as tumors have some hallmarks of primary fusion lymphoma gene expression, more this plasmablastic differentiation. KSHV co-infection drives activated effector and central memory CD8 T cell expansions. We also observe some IgM responses that are targeted actually against similar KSHV proteins is also seen in newly infected individuals, human individuals, especially in KSHV and EBV co-infected mice that cannot activate lytic EBV infection, reducing thereby EBV antigens. We observe T cell responses that preferentially recognize double infected B cells or presumably primarily directed against KSHV. We've identified 4 specificities of 44, of 66, of 73, and K6. Could so far only identify K6 also at the clonal level. We are now rescuing T cell receptors from these different specificities to compare them side by side during infection. And we can demonstrate that some of these T cell specificities have characteristics that could suggest that they are protective, such as the CD8 T cell clones against K6 that seem to be able to target EBV and KSHV double infected B cells, both in vitro and in vivo. So with that, what I would like to finish and thank the people in my lab that primarily did this work. So the double infection originally was started by Donald McHugh and has been continued by Nicole Cadeau. They are now followed up for the T cell, KSHV specific T cell studies by Lisa Rieble and Michel Böhni in my laboratory. It helped from a lot of collaborators, Denis Whitby for the specificity studies, both for antibodies and T cells, Jie Jung and Thomas Schulz for KSHV, recombinant KSHV viruses that we are currently exploring. Wolfgang Hammerschmidt in Munich for recombinant Epstein-Barr viruses, including the BCLF1 knockout virus. I would also like to thank these funding agencies down here for their support of our work and you for your attention. And I guess we will take questions and after the next two presentations. Thank you very much.
Yeah, thank you, Dr. Münz. Beautiful presentation. So we move to our next speaker, Dr. Lee. Dr. Lee is in the laboratory of an old friend of mine, Don Cleveland. Don and I, my name is Don. We were together with Mark Kirschner in prehistory at UCSF in a building that is now mainly medical, but was biomedical, actually biochemistry and biophysics when we interacted. But enough of history, let's talk about Dr. Lee. So Dr. Lee is a Damon Runyon postdoctoral fellow, quite an achievement. And she, of course, is in the laboratory of my friend Don Cleveland at UCSD, in the department of which he is chair. So Dr. Lee got her Ph.D. Not far from UCSD at Scripps. She's smart about where she hangs out. And she has her focus of her work is the poorly understood role of breakpoints of repeat DNA and initiating genomic instability and driving human disease. She has given presentations at numerous international conferences, and she will be presenting the talk, A Previously Missing Link between Epstein-Barr virus and cancer. Thank you, Dr. Lee. We look forward to it.
Thank you for that very kind introduction. And I also want to thank the Global Oncology Initiative for putting together this wonderful symposium. I'm definitely very honored and excited to be here to tell you about this, my work that is a previously missing link between Epstein-Barr virus and cancer. All right. Okay, so as you have already heard, and this audience is very, very familiar with, EBV is the first tumorigenic virus that was originally discovered in the 60s and is actually known for its ability to immortalize cells in culture. So it's an oncogenic virus. And EBV actually infects most of us in the human population, where latent infection after primary infection is thought to persist for a lifetime. And that is in the form of these extrachromosomal circular EBV genomes, so not integrated into the human genome. So they're extrachromosomal circular EBV genomes. And while obviously harmless in most people, EBV is linked to cancer, which is the theme of this session. And as you've already seen before, it's detected in up to 230,000 new cases of cancer worldwide every year. Although the exact link between EBV and cancer is somewhat unclear, and that's kind of the paradox of these tumor viruses, why we're all infected with the virus, but some of us will develop cancer while others won't. And so there are also, is there a laser point? Oh, here. Yeah, so cancer types that are strongly associated with EBV versus cancer types and diseases that are that have somewhat of a controversial association. So basically, it's kind of unclear exactly what the role of EBV is in tumor genesis. Nonetheless, in 2015, one of 7 cancer grant challenges put out by Cancer Research UK as well as the NCI is to obviously eradicate EBV cancers. So it's very important for global health. Okay. So now I want to zoom in on this protein called EBNA1, which you've already heard in some of the previous sessions. It is the only viral protein that's expressed in all latently infected tumors, regardless of the tumor type, regardless of the latency program. And that's because EBNA1 is sort of a bivalent tether that's essential for the persistence of these extrachromosomal EBV genomes in our human nucleus. Since these EBV genomes are not integrated into our chromosomes, they kind of have to have a mechanism of staying inside our nucleus as our cells divide. And that's through this EBNA1 protein glue. So just a little bit more about how this protein does that. So it has a C-terminal DNA binding domain that's able to bind to this 18 base pair palindromic sequence that's in the EBV genome. And it does that by forming a dimer where each EBNA1 molecule binds to half of the palindromic sequence. And this sequence is repeated about 20 times, or exactly 20 times, in a 2 kb region in the EBV genome. And EBNA1 also has N-terminal domains that can glue to our chromosomes in a non-sequence specific manner. So as you can see, this is exactly how EBNA1 can physically attach this circular EBV genomes to our human chromosomes, enabling DNA replication and also segregation of the EBV episomes with each cell cycle division. So if you use a fluorescent probe against either the EBV DNA or against the EBNA1 protein, you can see very nice colocalizing signal that's all over our nucleus at multiple locations. So it's not hard to see why EBNA1 has been extensively studied for its roles in genomic instability and cancer, both indirectly, just simply for enabling the persistence of the latent EBV genomes for latent infection. and also some direct roles that have been proposed, like the fact that some chromosome binding sites have been found to promote cellular survival, and it's known to dampen the DNA damage response and also interfere with cell cycle checkpoints. So then the question is, what else? It can't be that this is everything that EBNA1 does, and it's not completely understood. So what I wanted to know is maybe there are some unknown links between EBNA1 that's encoded by EBV and genomic instability in cancer. So as an initial experiment, I simply expressed a EBNA1 plasmid to express this protein in non-EBV infected cell lines that I routinely culture, since we are not a EBV focused lab and we don't study virology. So in Don's lab, we're interested in chromosomes and DNA and breakpoints and genomic instability. So that's why we routinely culture cells that are not infected with EBV. And this experiment led to a very striking and unexpected initial observation that led to this entire discovery. So what I saw was that EBNA1 forms a single pair of dots, basically dots, in the nuclei of non-EBV infected human cells that are diploid, for example, RPE cells and DLD1 cells. You can see that in each nucleus, there's a single pair of dots, and then also HeLa cells as well. And in U.S.A cells that are known to be pseudotetraploid, since they're cancer cells that have very messed up chromosomes, up to maybe 3 to 4 foci in each nucleus were observed. And this seemed to be specific to human cells, since no foci were observed in mouse cells. So as someone who, myself, I'm trained in sequence-specific DNA binding proteins. So when I saw this kind of foci formation that's formed by EBNA1, that's known to be a DNA sequence-specific binding protein, I knew immediately that this meant that these dots are actually marking clusters of the recognition sequence that EBNA1 is known to bind, which, just to remind you, is this 18 base pair palindromic sequence that's in the EBV genome, okay? So then, since these are human cells that are not infected with EBV, so they don't have any EBV DNA. So I thought that this observation meant that these foci must be labeling a cluster of EBV-like 18 base pair palindromic sequences in our human genome. And that's the only explanation for why I'm seeing this single pair of dots. So I searched the published literature, and there have been 903 EBNA1 binding sites that have been mapped across our genome, where all these dots are in EBV infected cells. And I wanted to see if there's maybe a cluster that can explain the foci that I was seeing. And indeed, this paper found that there is a cluster of EBNA1 binding sites on chromosome 11 at 11q23, although the word palindrome doesn't appear anywhere in this paper, and this site doesn't seem to be the focus of the paper. So I just looked more closely at the results in the paper. And that was when I saw that the sequence that's been found to be the binding motif at this cluster on chromosome 11, the very end portion of this sequence resembles exactly half of the palindromic sequence that EBNA1 has been known to bind in the EBV genome. So this was when I downloaded the actual genomic sequence at this cluster, and I simply did control find using my own, very basic bioinformatics tools. And this was the moment I saw that this GGG-TAAC sequence appears as a 18 base pair palindromic sequence that's repeated about 500 times in a 20 kb region on our human chromosome at 11q23. So hopefully you can appreciate. Right. Okay. So then next, to validate that this indeed is the location of the foci that I initially observed, I used a labeling approach using nuclease dead Cas9. And what I saw was that a MIC tagged EBNA1 in red here colocalizes perfectly with a flag tagged nuclease dead Cas9, whereas a MIG-tagged EBNA1 that's red here does not colocalize with a flag dCas9 that's marking a separate cluster. So this flag dCas9 is designed to target the cluster at 11q23 that I just previously showed you, that I hypothesized to be the location of the cluster. And then furthermore, I used a Cas9 mediated cutting approach to see if whether targeting the actual 18 base pair palindromic sequence to eliminate the sequence would eliminate foci formation, which then would again suggest that this 11q23 position is the location of the foci that I initially observed. And that's exactly what I saw. So using a guide RNA that is a control guide RNA, there is no there's no decrease in foci formation. So you can still see the pair of foci in each nucleus. And then using a guide RNA that targets the 18 base pair palindromic sequence at 11q23, I was able to see that the foci formation was basically abolished. And these data suggest that the cluster at 11q23 is indeed the location of the EBNA1 foci that I observed. So here is a summary of what I've shown you so far, which is that our chromosome 11q23 contains a cluster of EBV-like 18 base pair palindromic sequences. So you can see that the EBV palindrome looks like this, which is somewhat of a perfect palindrome, but our human palindrome looks slightly different in the sense that the central 4 base pairs are non-palindromic. And then there are also other differences. For example, in the EBV genome, there are 20 copies of this palindromic sequence. And this palindromic sequence appears in a 30 base pair repeat unit. Whereas in the human genome, there's about 500 copies, so a lot more, in a longer region on our chromosome 11. And this 18 base pair palindromic sequence appears as part of a 42 base pair repeat unit that's repeated 500 times. Very interesting. And so bioinformatics search for the human palindromic sequence does indeed only find one unique peak across our entire genome, which is consistent with the single pair of foci that I initially observed. And just a little bit more about this cluster in terms of evolution, whether it's evolutionarily conserved. We found that it is conserved amongst humans, gorillas, chimpanzees, gibbons, and that's about it. So only these 4 organisms. And again, it's not even in mice or anything else. And here you can see a zoomed in version of the sequence that's at this repetitive site. And between the human and the chimpanzee, the sequence is pretty much identical. And then here's the EBV one where you can see some of the differences. Okay, so now you might be wondering what exactly is the significance of all of this and why do we care that there is a cluster like this in our human genome? So there's this emerging link between repeat DNA and genomic instability in that repeats in our genome are known to be hotspots of DNA damage, which again is obviously a problem for the genome. And that's that's due to the fact that repeat DNA has the unique ability to form long tracks of alternative either DNA structures or DNA protein structures. And these structures are known to impede anything that is chromatin associated. So for example, transcription machinery or replication machinery they cannot pass through sections where you get long tracks of alternative structures. And what that would do is break the DNA, and that results in genetic instability. And so these mechanisms, since I've drawn them like this here, but in reality, they're very poorly understood because repeat DNA is overall very poorly understood in the field of genomic instability. But it's not surprising that repeat DNA is a characteristic feature of these breakage-prone regions in our genome called fragile sites. And why do we care about fragile sites? Because they correlate with recurrent breakpoints in cancer genomes. and they're thought to enable structural variants of chromosomes that can drive tumorigenesis. So basically, from a normal genome, you get breakage of these fragile sites. So breakage of fragile sites can turn a normal genome into a cancer genome that is very messed up with all kinds of chromosome abnormalities. And that's because a single chromosomal break is actually very powerful in terms of fragmenting the entire chromosome. So you end up with a centric piece of the chromosomal fragment that can form dicentrics, which can then missegregate into micronuclei during cell division. And you can also have the smaller acentric piece that gets immediately missegregated into something called a micronuclei. And the micronucleus is the site of something called chromosome shattering or chromothripsis that can generate additional breakpoints anywhere along the rest of the chromosome. So that's how you can get from a single breakpoint on the chromosome to all kinds of chromosomal abnormalities along the rest of the chromosome. So chromosomal fragile sites have been studied since the 1980s. And right now, I think there's been about 100 or so sites that have been discovered. And of course, 11q23 is one of the fragile sites that have been mapped in our genome. And that is precisely where the EBV encoded EBNA1 protein binds at the cluster of EBV-like repeats. So our next prediction was that EBNA1 binding to these palindromic repeats would result in breakage. Okay, so just to elaborate on the model that we're proposing here before I show you the evidence for it. Basically, normal DNA that's non-repetitive is able to replicate and then condense onto mitotic chromosomes and segregate properly, no problem. Repetitive DNA can form alternative structures that are kind of difficult to replicate. So they might form abnormal structures that are that undergo abnormal condensation on mitotic chromosomes, but everything is still intact. So everything can segregate properly during mitosis and be inherited properly. Because after all, repeat DNA is still part of our normal genome. However, if you have something like EBNA1 that now binds here at a single cluster that's very spatially, a cluster of EBNA1 bound to these repeat sequences, then you can get, you can push everything sort of over the thresholds of what's okay, and then that induces breakage, generating these very unstable mitotic structures that can then fragment the rest of chromosome 11. So to see whether this was happening, I used a fluorescent probe against a portion of the actual repeat unit. And what I saw was that in cells where I was able to induce EBNA1 expression, this repetitive site marked by the fluorescent foci, it does indeed colocalize at either a gap on the centric chromosome or on highly aberrant small chromosomal fragments, indicating that breakage and then aberrant fusion has occurred. And then also when I looked at interphase nuclei, I was able to see that this repetitive site does indeed get missegregated into these micronuclei that are a structure that's induced by chromosomal breakage. And next, to further understand what's happening to the rest of the chromosome 11 fragments beyond just the repetitive site that I was able to mark, what I did was a dual colored fish approach using a green probe against the entire chromosome 11, and then a red probe against either the ATM tumor suppressor gene that is immediately proximal to the site of EBNA1 binding, or a red paint against the mixed lineage leukemia proto-oncogene that is rearranged in 10% of leukemias and up to 90%. And yes, the EBNA1 binding site is exactly situated between major ATM tumor suppressor and the mixed lineage leukemia proto-oncogene. So then what I saw was exactly what I expected, which is that using the ATM red probe, the break is immediately distal to the ATM gene. And I was able to see that ATM does indeed get missegregated into these micronuclei structures. And then also mixed lineage leukemia gene is indeed on the small acentric distal piece after induced EBNA1 expression. And it also ends up in the micronuclei structures. And again, just to remind you, micronucleus is the site of chromosome shattering or chromothripsis that can fragment the rest of chromosome 11, so anything that gets into micronucleus. So at this point, I just want to point out that ATM is a major tumor suppressor that is involved, it plays a central role in the DNA damage response pathways. So from this discovery, I think that it suggests that EBV, dependent breakage at 11q23, can indeed trigger inactivation of a major tumor suppressor like ATM. And then, like I said, the mixed lineage leukemia proto-oncogene translocations of this proto-oncogene are found in a lot of leukemias. And now perhaps this EBV-dependent breakage at 11q23 is an initiating event that can explain how MLL can be broken and translocated into the MLL proto-oncogene and then expressed into the MLL fusion oncogenic protein. Okay, so far everything I've shown you, the evidence has been from non-EBV infected cells, which is where this entire discovery initially took place, because I don't study EBV and we're not an EBV lab. But the important next question is, what about in the EBV infected cells that are truly infected with latent genomes? So it turns out that in this setting where I've shown you, increasing levels of EBNA1 binding can trigger dose-dependent 11q. So below a certain threshold of EBNA1 binding, there is no breakage. And the more EBNA1 you express, the higher frequency of breakage I was able to detect. So in terms of thinking about now EBV infected cell lines, my question was, how can these infected cell lines survive this kind of crazy rampant breakage of chromosome 11? Like, that is very toxic. It cannot be good for cells. So then maybe latent expression of EBNA1 that's maintained at very low baseline levels is below the threshold sufficient for breakage. And that is exactly what I saw, that latently infected cell lines express EBNA1 at approximately half the lowest EBNA1 level that's required to induce breakage. And when I induced about a doubling of the EBNA1 levels that are normally in the latently infected cells, I was able to detect 11q23 breakage in cells. So then next we looked at actual cancer genomes to see if we can find evidence of this mechanism in cancer whole genome sequencing. And indeed, we saw that chromosome 11 has the most structural variants in EBV-associated nasopharyngeal carcinoma compared to other chromosomes, including translocations of chromosome 11, particularly with chromosome 3. We're not sure why yet, but something very interesting. So then also in 63 chromosome 11 rearranged nasopharyngeal carcinomas, we were able to find these clustered rearrangements in about half of them that are consistent with what you would expect from the kind of fragmentation that's taking place in the micronucleus that's called chromosome shattering or chromothripsis. So then coming back to this model that I'm presenting, what this means, what I think this means is that in latently infected cells, any kind of transient burst of EBNA1 levels above the threshold that it's normally maintained at is able to trigger breakage and fragmentation of chromosome 11, which then would drive genomic instability, chaos, which is obviously a hallmark of cancer. And this kind of burst of EBNA1 levels is relevant for settings like any kind of epigenetic de-repression of transcription that might take place during reactivation of EBV, which is what we think would be the physiological setting that this mechanism is important for. And going forward, this is a previously missing link between repeat DNA as well as genomic instability, as well as viruses. This kind of site-specific breakage induced by a single protein encoded by EBV at a single cluster of repeats in our genome. And this kind of breakage of repeats can set off local repeat instability, which can turn normal repeats into repeat expansion. And repeat expansion is at the root of a set of diseases called repeat expansion diseases, including ALS and frontotemporal dementia and Huntington's disease. And also, on the other hand, anytime you have breakage of DNA, you obviously have chromosome instability, which is a hallmark of cancer. So I think that this kind of discovery is a very fresh and exciting opportunity to really understand the breakage of repeats that's important for all these humans. In the future, I would really like to capitalize on this discovery of site-specific fragility induced by EBNA1 at repeats to understand exactly how broken repeats can threaten a healthy genome and drive these human diseases, particularly in these virally infected cells. So things like how exactly the breakage happens, the consequences, and the triggers that provoke breakage, and also obviously clinical applications. So what are the actionable items that can come out of this kind of discovery for EBV-associated cancer? And then, so zooming out, like a bigger picture question would be, are there other tumor viruses with this kind of similar mechanism for HPV, maybe, or for KSHV? And also, other than the fact that this mechanism is inducing breakage of the chromosome when there's too much binding, what is the biological function of having a cluster of these virus-like repeats in our genome? You know, because the function cannot be to break our DNA. It doesn't benefit the virus, it doesn't benefit the host. So what is the actual function of this kind of binding, other than the fact that it creates problem in. And with that, I would like to acknowledge my advisor, mentor, Don Cleveland, and all the members of the Cleveland Lab, and also my long-term collaborator, Ludmil Alexandrov, and his grad student who performed all the computational and bioinformatics analyses that made this work. And it's been really exciting sharing discovery with them. And thank you for your time.
Well, I don't think we have to worry about the next generation of scientists if they're even a fraction of what Dr. Lee presented to us. So thank you very much. So we're going to turn to our final speaker of this session, Dr. Jeff Cohen. He'll be speaking from Bethesda, I believe, possibly Rockville. He is well known in the EBV field, and he will be discussing a number of very important issues regarding developing of therapeutics, prophylactics against EBV infection. So Dr. Cohen received his degree from Johns Hopkins and his medical degree, and he was a resident at Duke. Following a fellowship at NIH, he was a clinical fellow at the Brigham and an instructor in medicine as well. He returned to NIH and became the chief of the medical virology section in the Laboratory of Clinical Infectious Diseases until 2010. And then Dr. Cohen became chief of the Laboratory of Infectious Diseases in the same locale. He's been decorated with many honors and memberships of various clinical and scientific societies. And importantly, he's been the recipient of the NIH Directors Award and the NIAID Merit Award. So his topic today is entitled Use of Vaccines and Monoclonal Antibodies to prevent Epstein-Barr virus infection and disease. We welcome you virtually, Dr. Cohen. Thank you.
Thank you. I hope you can see my screen. So in terms of disclosures, I'm a named inventor on U.S. government patents on EBV vaccines and monoclonal antibodies. So we heard this morning that EBV is associated with up to over 200,000 malignancies each year worldwide. And it turns out that gastric carcinoma is actually the most common EBV malignancy. We heard from Dr. Lowy that gastric carcinomas are the 5th most common cancer in men and fourth most common cancer in men and 5th most common cancer in women. And about 8 to 9% of gastric carcinomas are EBV associated for a total of 84,000 cases per year. Nasopharyngeal carcinoma is particularly common in Southeast Asia and parts of North Africa with 74,000 cases per year. Hodgkin's lymphoma, 29,000 cases, and Burkitt lymphoma, particularly in sub-Saharan Africa, at 7,000 cases per year. There are other EBV-associated malignancies, including non-Hodgkin's lymphoma, and we heard from Dr. Kopal about extranodal NKT cell lymphoma and plasmablastic lymphomas, which are EBV-associated. It also is associated with lymphoproliferative disease, either post-transplant or individuals with primary immunodeficiency. And as you all know, it's the major cause of infectious mononucleosis. And Dr. Münz mentioned the association of EBV with multiple sclerosis. So in terms of trying to develop a vaccine for Epstein-Barr virus, the virus encodes a number of glycoproteins, which are important both for entering B cells and glycoproteins important for entering epithelial cells. Most of the work prior to the last few years is focused on GP350, the major glycoprotein on the surface of virus-infected cells. And GP350 binds to its receptor, CR2 or CD21. Dr. Agembo has shown that it also binds to CD35. GHGL, GP42 form a trimeric complex on the surface of B cells, and GP42 binds to HLA class II. This activity actually results in activation of GB, and these glycoproteins are important for fusion of virus to B cells. And again, GP350 is important for attachment of virus to B cells. The virus infects the epithelium in the back of our throat, and BMRF2 is another viral protein that attaches to epithelial cells. And for fusion for epithelial cells, GH and GL interact with Ephrin A or A2 or integrins. And this presumably induces a conformational change in GB, again, which is important for fusion of the virus to epithelial cells. So as I mentioned, GP350 has been the major target of virus vaccines. And that's because GP350 is the principal target for neutralizing antibodies to prevent B cell infection. It's also a target for antibody-dependent cellular cytotoxicity. And it's also a target for CD4 and CD8 cells. So a number of years ago, a company developed a soluble form of GP350 and tested it in a phase 2 randomized double-blind placebo-controlled trial. In this study, 90 individuals received the vaccine, 91 received placebo. They were all seronegative prior to vaccination. And you can see these individuals got the vaccine at 0, 1, and 5 months. And what you can see here is that individuals who got the vaccine, shown by these black circles here, those individuals had a 78% reduction in mononucleosis compared to individuals who got the placebo. However, it's important to note that the individuals who got the vaccine did not have any lower rates of infection by EBV. So again, this vaccine reduced the rate of mononucleosis, but did not reduce infection by EBV. And presumably for this reason, the vaccine was not further developed. So we asked the question whether we could improve upon soluble EBV And to that end, we have been using nanoparticle-based vaccines. And you heard from Dr. Lowy this morning that the HPV vaccine is contains a multivalent, symmetrical, repetitive array of antigen, and it induces an incredible immune response such that individuals have broadly or long-lasting antibody responses to HPV. The hepatitis B vaccine also is a nanoparticle vaccine, which is highly effective. So we collaborated with our colleagues at the Vaccine Research Center here at NIH and used a molecule called ferritin. And we used the ferritin from Helicobacter pylori, which is very different in amino acid sequence from the human ferritin. But like its human component, it self-assembles into a 20-former complex shown by this soccer ball-like appearance here. And the folks at the Vaccine Research Center had put influenza HA covalently bound to ferritin and found that this nanoparticle induced very high titer, broadly neutralizing antibodies in mice. And this vaccine has been in 2 clinical studies thus far. So we engineered GP350 onto each copy of ferritin to develop these nanoparticles. And shown here is GP350 with its ectodomain or extracellular domain, a transmembrane domain, and a very short carboxy terminus here. We made a similar construct to what was in the clinical trial vaccine, that is soluble GP350, although we added a his tag just to help purify it. And we made a number of GP350 ferritin constructs, and the one that worked the best contained domains 1, 2, and 3 of GP350 fused to ferritin. And domains 1, 2, and 3, shown on the top, contain the receptor binding site for GP350, the CR2 binding site. So we transfect cells with either ferritin alone or GP350 ferritin. And what you can see here is that the GP350 ferritin constructs have the central ferritin core, but have GP350 decorating the central core. And so you can see the glycoproteins radiating out from ferritin. And we immunized mice with either the soluble GP350 ferritin, similar to what was in the human trial, or our GP350 ferritin. And we look at the neutralizing titer for B cells after vaccination. These mice, you can see, got vaccinated at 0, 3, and 16 weeks. And what we found was that the mice that got the GP350 ferritin had 100 to 1,000-fold higher neutralizing titers for EBV compared to the animals that got the soluble GP350. So it looked like it was much more potent than soluble GP350. So why might this be more effective? We collaborated with the late Michael Rossman at Purdue and did cryo-electron microscopy on our GP350 ferritin molecules. And what you can see here is a vaccine particle, a nanoparticle, and shown in pink is the CR2 binding site, the receptor binding site for GP350. And the distance here is 50 to 100 angstroms, which is precisely the optimal distance to cross-link B cell receptors. So we think that this molecule displays the neutralizing epitope of GP350, or the primary neutralizing epitope of GP350, optimally to induce an optimal antibody response. And I don't have time to show it today, but we found that when we immunize monkeys with this vaccine, 55% of the antibody that they make is directed to the CR2 binding site. Whereas if we immunize monkeys with the soluble form of GP350, only 5% of the antibody they make is directed to the CR2 binding site. So this display shown here really helps to focus the immune system on the most potent sites for neutralizing EBV through GP350 antibodies. So to that end, we have started a clinical trial of GP350 ferritin here at the NIH. We've vaccinated either EBV seronegative individuals or seropositive individuals, young adults, and they've received 3 doses of GP350 ferritin in matrix M adjuvant. This is the adjuvant that used in Novavax's SARS-CoV-2 vaccine and contains saponin. We've been vaccinating these young adults at 0, 1, and 6 months, and we'll be following up 1 year after the last dose. The primary endpoints for our clinical study, of course, it's a phase 1 study, so it's safety, either local at the site of injection, systemic symptoms, or other reactions. And we also will be looking at the change in EBV neutralizing antibody titers between baseline and 1 month after the 3rd dose of vaccine. Secondary endpoints are GP350 specific antibodies, as well as CD4 T cell responses to GP350. So what I've mentioned so far is a GP350 vaccine, but you've seen this before, and there are other glycoproteins important for EBV infection. So to try to figure out what their relative contributions were to preventing EBV infection, in our laboratory, we made cells that expressed either GP350 or GHGL or GP42, and then incubated these cells with human plasma to deplete all the antibody that recognizes GP350 and see what amount of neutralizing antibody was left, or depleted the antibody to GHGL or the antibody to GP42. And what we found was that about 50 to 60% of the neutralizing antibody in human plasma is directed against GP350, and 20 to 30% is directed against GHGL, and pretty much the remainder against GP42. So if we had these 3 immunogens in our vaccine, we would really presumably have what was really needed to neutralize B cells. For epithelial cells, GHGL constituted 75 to 80% of the neutralizing antibody to prevent EBV infection of epithelial cells in human plasma. So this suggests that EBV vaccines targeting GP350 and GHGL GP42 should induce antibodies to neutralize infection of both B cells and epithelial cells and might prevent infection and disease. So to that end, we've made nanoparticles that have either GHGL on them or GHGL and GP42 on them. And again, this was done by fusing GH to ferritin, this time the full ectodomain of GH fused to ferritin. And if we transfect cells with this GH ferritin construct in GL, we get GHGL ferritin nanoparticles, or if we transfect cells with GH ferritin, GL, and soluble GP42, we get GHGL GP42 nanoparticles. So we went ahead and compared the immune responses in mice to GP350 ferritin or GHGL GP42 ferritin, or the combination, to think about what next generation vaccine for us would be best. And what you can see is that when you combine GP350 ferritin with GHGL GP42 ferritin, you get about a 5 to 6-fold increase in neutralizing titers for B cells compared to GP350 alone, and a very slight increase in terms of neutralizing antibodies to GHGL GP42. In terms of epithelial cells, GP350, as we predicted, really doesn't have very much neutralizing activity, or doesn't induce very much neutralizing activity for epithelial cells, but GHGL GP42 does. So with that in mind, and again, what I've shown you so far is just antibody responses to these vaccines. And the question is, do you really get protection from infection or disease with such vaccines? So EBV only infects human lymphocytes or epithelial cells. It does infect some non-human primate cells, but it doesn't induce malignancies in non-human primates, except at very, very, very high levels. However, humanized mice, which Dr. Münz mentioned already, are immunocompromised mice. They're irradiated, and they're given human CD34 positive stem cells. So we take these humanized mice, which have human stem cells, which differentiate into human lymphocytes, and we give them purified IgG from vaccinated mice intraperitoneally. And then we can challenge them with EBV intravenously and measure survival, viremia, EBV infiltration of tissues, EBV lymphoproliferative disease, and actually EBV lymphomas. So again, these mice, we take normal mice, that is non-immunocompromised mice, vaccinate them with either GP350 ferritin, GHGL ferritin, or GP350 ferritin plus GHGL GP42 ferritin. We collect serum from the mice, purify the IgG, and then give that IgG to humanized mice at day -1, 0, +1, and then challenge them with EBV intravenously. In the control mice that got IgG from naive mice, you can see the control mice develop viremia at week 7 and week 9 after challenge. And most of the mice actually develop viremia even at week 5. However, the animals that got the GP350 ferritin plus GHGL ferritin, or the animals that got GP350 ferritin plus GHGL GP42 ferritin, were largely protected from viremia. In each case, only one animal was viremic. And when we look at the tissues from these animals, liver, spleen, or kidney, for EBV infected B cells by staining for Epstein-Barr virus encoded RNA, you can see the animals that got the IgG from the control cells and then from the control mice and then were challenged with EBV. Most of them had EBV infected lymphocytes in their tissues, but the animals that got IgG from the vaccinated mice really did not have evidence of EBV infected cells in their tissues. Now, when we looked at the tissues from the animals more closely, you can see that the control animals, about 50% of them developed EBV lymphomas. These are shown here on H&E. They're human B cell positive, they're CD20 positive, and they contain EBV encoded RNA, which are these purple dots here. But the animals that got IgG from animals that were vaccinated were completely protected from EBV lymphomas. So we are now planning to do a second generation vaccine trial in which we'll give humans either nanoparticles for GP350 plus GHGL GP42, or not shown here, but GHGL GP42 alone. And we hope to start this trial the first quarter of 2024. Moderna already has a vaccine trial ongoing in which they vaccinate individuals with 4 separate RNAs at the same time for GP350, GHGL, and GP42. And there's no results from that trial as of yet. So I don't want to be remiss without mentioning there are other EBV vaccines in preclinical trials right now. In the 1980s and 1990s, GP350 was tested by several individuals at Bristol in England, either the native form of the protein, recombinant protein, GP350 from an adenovirus vector from vaccinia. And this protected cotton-tailed tamarins from lymphoma. However, these animals are an endangered species and really can't be used at this time. But it also reduced virus replication in marmosets. There's another vaccine that's been underway, which is an EBV virus-like particle in which they've deleted LMP1, latent membrane protein 1, Epstein-Barr virus nuclear antigen 3s, and BZLF1. And this induced both neutralizing antibody and cellular immune responses. A group at U.S.A. Here in Bethesda had a trimeric GB and GHGL vaccine in alum and CpG, which also induced neutralizing antibodies and protected animals by passive transfer. And then Dr. Agembo's group at City of Hope, of course, have developed a Newcastle disease virus-like particle that is actually has GP350, GHGL, GP42, and GB. So this pentameric particle in alum and NPL, and induced really potent neutralizing antibodies as well. So what might be the outcome of an EBV vaccine? Well, you know, the varicella vaccine, which is the only vaccine we have to prevent a herpes virus infection, does not induce sterilizing immunity. And it's possible that an EBV vaccine might not induce sterilizing immunity. However, it might limit the level of replication and expansion of T cells during initial infection. And mononucleosis, the symptoms of mono, are actually not due to EBV, but due to the expansion of T cells, as well as some NK cells in response to the infection. So a vaccine might reduce mono, and a vaccine might also reduce the viral load if it controls the initial infection. And it's been shown many times that the higher the viral load in transplant recipients, the higher risk of developing post-transplant lymphoproliferative disease. And then finally, a vaccine that inhibits or reduces mononucleosis might reduce EBV-associated malignancies such as Hodgkin's lymphoma. And another group has shown that individuals who have infectious mononucleosis are at higher rate for EBV Hodgkin's lymphoma during the first 9 or 10 years after in mononucleosis. So even a vaccine that did not induce sterilizing immunity could still reduce the rate of certain EBV malignancies, such as EBV Hodgkin's lymphoma. So potential vaccine trials: prevention of infectious mononucleosis in college-age students, prevention of post-transplant lymphoma in seronegative persons about to receive an organ or stem cell transplant, prevention of EBV disease in seronegative boys with X-linked lymphoproliferative disease, and then prevention of other EBV diseases like multiple sclerosis, Hodgkin's lymphoma, Burkitt lymphoma, nasopharyngeal carcinoma. One would need very large studies to do these vaccine trials. And it's more likely, I think, that a vaccine would be licensed to prevent mononucleosis, and then phase 4 studies after licensure, following up individuals who got vaccinated versus who didn't, and looking at the rates of these malignancies that have long latency periods might be the way to go to see if a vaccine can prevent these types of malignancies. Now, highly immunocompromised persons, such as transplant recipients, might not respond to an EBV vaccine. They just may not have a good antibody or good T cell responses. And it's known that EBV seronegative transplant recipients have much higher rates of developing post-transplant lymphoma or lymphoproliferative disease than seropositive individuals. So if they don't respond to vaccines, perhaps one could use monoclonal antibodies to EBV that might protect these individuals from infection or disease, and might reduce the rate of lymphoma after transplant in these seronegative individuals by keeping the viral load low, which, as I mentioned, the viral load is a risk factor for developing PTLD or lymphoma. And similarly, patients with X-linked lymphoproliferative disease or other immunodeficiencies are at high risk for developing fulminant fatal infectious EBV disease with primary infection. Some of these have been given IVIG prior to infection, but breakthroughs and fatal breakthroughs have actually occurred. So there might be a role for monoclonal antibodies in these immunocompromised individuals. So we have looked at monoclonal antibodies. We've developed them to GP350, GHGL, GP42, and GB. And to date, the ones that have been most effective in our models have been monoclonal antibodies to GHGL or GP42. And as an example, I'm going to show you a monoclonal antibody that we developed to GHGL. So we took blood bank donors at the NIH and looked at individuals who had the very highest EBV neutralizing titers, and then isolated monoclonal antibodies from the blood of those with the highest neutralizing titers. These are just a few of the GH monoclonal antibodies that we developed. And what you can see is the antibodies that need the fewest micrograms per mil to neutralize B-cell infection, that is this 769B10, are the most potent for protecting B cells from infection, or it's also the most potent for protecting epithelial cells from infection. And in terms of neutralizing activity, you can see again, B10 has the, you need the lowest amount of micrograms per mil to prevent infection of B cells or epithelial cells. And it has a binding affinity in the picomolar range. We've done crystallography of these monoclonal antibodies bound to GHGL in collaboration with Gordon Joyce. And we found that these antibodies bind to different portions on GHGL. And the most potent antibody, B10, binds to GH here, really right near the site where it binds to, where GH binds to GL. So we tested these monoclonal antibodies in our humanized mouse model. And again, when you infect the animals and just give them, if you give them PBS ahead of time and infect the animals, they all become viremic. If you give them the one GH monoclonal antibody C2, they all become viremic, or F7. But with B10, only one of the animals became viremic. And again, when we look at tissues from these animals, lung, liver, spleen, or kidney, B10 protected all but one of the animals from having EBV-infected lymphocytes in the tissues. And when we looked at macroscopic lesions, atypical lymphotraits, lymphoproliferative disease, lymphoma, or the combination of them, only the B10 monoclonal antibody completely protected the animals from lymphomas or lymphoproliferative disease. So to summarize, EBV GP350 vaccine reduces EBV mononucleosis, but did not prevent infection. Again, this was the soluble GP350 used in the phase 2 clinical trial. We think the ferritin vaccines, by having a more potent GP350 that's cross-linking B-cell receptors, can quantitatively give us better neutralizing antibody, but by including GH-gel GP42, will qualitatively give us better antibody by having additional epitopes represented in the vaccine. Sera from mice immunized with the nanoparticle vaccines protected humanized mice from viremia, organ infiltration of virus-infected B cells, and lymphomas. And a GP350 nanoparticle vaccine and GP350 GH-gel GP42 mRNA vaccines are already ongoing in clinical trials. Monoclonal antibodies to GH-gel, and I didn't have time to show today, but also to GP42, neutralize EBV infection, block fusion, protect humanized mice from death, viremia, EBV infiltration, and B-cell lymphoma. And such monoclonal antibodies might be used to prevent infection or disease in transplant recipients or immunocompromised persons who might not respond well to vaccines. So this work was done by a very talented staff scientist, Wei Bu in my lab, and by a graduate student and several postbacks who worked over the years on this project. Dr. Dirkie Schock is running the clinical trial. Dr. Marco Trigiani has helped us with some structural studies, and it's been a collaboration with the Vaccine Research Center. So I'll stop here, and presumably we can go back to the Q&A part of the meeting.
Now we transition to questions from the audience to the speakers, and I could probably fill up an hour, so I'll wait to hear from others first, and then I'll be happy to chime in.
Yeah, all great talks. Yeah, so for Dr. Lee, so really amazing work. I have two questions. The first part, it might just been me, I might have missed it. So I know you did a lot of informatic analysis looking at protein binding motifs. You did ChIP-seq, right? Where you? So
that was actually previously published.
Okay, so that was previously published. You didn't do it.
I just looked through their published data.
Got it. And then my second question is, like, many repeats, when you think about dinucleotide, trinucleotide repeats that are associated with fragile sites, there can be sort of differences in the length of the repeats. Is this a static length? Or if you looked across individuals, would you see, 40 repeat units on one allele, 20 on the other, or is it sort of static at the 40 or 42 or whatever? I can't remember.
Yeah, exactly. So that's definitely one of the interesting aspects of this discovery is that this repetitive site is indeed polymorphic in the human population. So it does show variation in terms of how many repeats we each have from individual to individual. And our evidence of supporting my statements comes from analysis of just 4 individuals. And it was pre-existing sequencing data that the bioinformatics graduate student looked through. And she was able to see a 2.5-fold difference in repeat copy number between 2 ethnicities. It was Chinese versus Jewish. So the Chinese individuals had 800 copies, whereas the Jewish individuals had about 300 copies. And then in the slides that I showed, I labeled everything as 500 copies because that's using genome assembly 38, which is the generic short read sequenced, assembly of our human genome that's in all the databases.
No, it's
So
fantastic. Could open up a really interesting line of investigation around
definitely.
susceptibility. Yeah.
Exactly,
Yeah, very
because
cool.
one of the problems with EBV associated cancers is, again, it's a paradox of why we're all infected, but only some of us will develop cancers. So this mechanism suggests that one aspect of susceptibility is how many copies of the repeat we have. The more copies, the more prone to breakage
Right.
the repetitive site is. Yeah, thank you.
Thank you.
Yeah.
Any other questions from the audience? Dr. Ogembo, you must have something. No? Go ahead.
Yes, my question is going to Dr. Cohen. Dr. Cohen, based on the explanation you gave in EBV entry, it seems that GB is playing a very crucial role for the entry in both B cell and epithelial cells. But it seems in many cases, many people are not using as a vaccine candidate. Why so? Because seems from the RSV, seems if you target the fusion mechanism, you basically can make an effective vaccine rather than targeting 5 different glycoproteins.
Yeah, so I think that's an excellent question. There was just a paper actually online in Cell Host and Microbe showing that a GB particle vaccine induces very potent immune responses. We've been more focused on trying to have a more pre-fusion type of GB, which you're alluding to for the RSV vaccine. And to date, haven't really been able to make a construct that is particularly potent for immune responses. But I think your point is very well taken, and that's sort of the next candidate to add to a prophylactic EBV vaccine. Because again, I think inhibiting fusion of the virus to cells is a really critical step for preventing infection of B cells and as well as epithelial cells.
Thank you. Yeah, I ran into Barney Graham and he was talking a little bit about a herpes GB pre-fusion, but he didn't tell me enough. So are there other questions? Go ahead.
Yes, I had a question about the nanoparticle vaccine. It's a ferritin, and I wondered if you were able to do biodistribution studies to track it by MRI, or is it too small?
I think it's too small. We have not done biodistribution studies.
Okay.
It's small enough that it's going to go through.
5 nanometers, pretty small. Yeah. Okay.
Yeah.
Thank you. So I have a question. It's either for Dr. Münz, who we haven't heard from recently, or Dr. Cohen. And we all in the field, the herpes virus field, anxiously await how Moderna's vaccine strategy will bear out. But based on what we know, and I think Dr. Lowy alluded to that, for SARS-CoV-2 and the fact that when we measure immunity several months later, we at least know that the neutralizing antibody titers drop precipitously over a 6 months period. Hence, Operation Next Gen, et cetera. We're constantly reformulating new vaccines. So EBV, as we know, is a lifelong chronic infection, very similar to my friend CMV. So how do you envision a vaccination strategy having sufficient durability, and which part of it would you imagine would be durable enough to A, prevent infection, the famous Stan Plotkin's term, sterilizing immunity, or prevention, at least in those who are already infected, who may become immunosuppressed, to prevent viremia. So either of the gentlemen would be of interest to hear what you have to say, or both.
Maybe I can give it a start and then Jeff can continue. So I guess, the question is really, as I think Jeff also alluded to, what does it take to convert an asymptomatic primary infection to an asymptomatic primary infection? Because that is most likely the scenario that most of these vaccine candidates will address first, right? And it could very well be that even though the RNA vaccines induce only short-lived or fairly short-lived plasma cells, right? That's the basis for the waning antibodies. That memory B cell responses are more durable, and also underlying T cell responses that help these B cell responses. And we will see, I guess, if that is sufficient, even after maybe plasma viral loads have waned, right, to still confer some protection against infectious mononucleosis. But I think at the moment it's probably difficult to predict.
You?
Yeah, I would say that we've been focusing on the nanoparticle approach, based a little bit on what Dr. Loeys mentioned this morning about just how potent and durable the immune responses have been to the human papillomavirus vaccine. We can't say that our EBV vaccine is going to be that durable, but we think that that might be an advantage over the mRNA vaccines. The one, to be fair, our vaccine is less likely to induce CD8 T cell responses. It doesn't get, inside the cell and processed like an mRNA expressing protein would be. So I think there are possibly advantages and disadvantages to both. But I think Dan has really hit the needle, really spot on in terms of the durability question, just because it's really important to have a vaccine that's going to be effective for a long period of time. And we've talked to some epidemiologists in England who point out that the most important factor for an EBV vaccine is going to be its durability, so that we don't have adults that are seeing EBV infection for the first time at an age when they're at higher risk for developing more serious EBV problems compared to children. So again, I think a vaccine would need to be used at a young age, and it's going to need to be durable, and maybe you'll need to have multiple boosts throughout life.
Thank you both, Dr. Münz and Dr. Cohen. Dr. Ogembo,
Yes,
go ahead.
I have a follow-up question for Dr. Münz. Based on your presentation from the humanized mouse study, it seems, if I did get it right, that you are showing that K6 could be a targetable for CD8 as a CD8 target for therapeutic vaccine. How does that correlate with the studies that has been done in epidemiology from human? Is that also the main target T-cell antigen or not? And also following up on that for Dr. Lee, EBNA1 is the only EBV protein expressed in all cancer cells. How targetable it is based on the fact that recently has been shown not only to be the protein needed for tethering, but also closely the antibody targeting EBNA1 is probably the causal of multiple sclerosis.
Maybe I can start again.
Yeah, go ahead. Why don't you start and then we'll move to Dr. Lee.
Perfect. Yeah, so K6 is indeed a minor T-cell antigen, right? So in Dennis Whitby's studies in Uganda, it has been only found to be the dominant T-cell antigen in one donor that was screened in that cohort. This being said, unfortunately, immune dominance in KSHV-specific T-cell responses is very poor to start with. The antigens that are indeed the most immunodominant, if you will, right, and are recognized in 20, maximally 30% of the KSHV carriers are K8.1 and LANA, right? So the very same that also the antibodies preferentially seem to recognize. So I guess at the moment it's unclear, right, if such a response that is very frequent in infected individuals, right? But as a vaccine target, I think it could still be an attractive target if we can demonstrate that it mediates actually protective effects, even though only a small subpopulation might actually make these immune responses.
Thank you. And Dr. Lee.
All right. Okay. So the second question was how targetable EVNO1 is. Now that this mechanism has come to light, can we do something about it to prevent it from happening? Especially because EVNO1 has been recently implicated in multiple sclerosis as well. So I think, yeah, so definitely one strategy would be to degrade EVNO1 somehow. And there are molecules that can that have been developed that can be repurposed to do that. I think that would be one strategy, is to degrade EVNO1. And then another strategy is to develop molecules that can bind the repeat sequence and outcompete EVNO1 binding there. And so there are molecules, for example, polyamide molecules that are known to bind repeat sequences. So you can imagine that you could develop one that would bind to this repetitive site and kind of protect it and mask it from EVNO1 binding. And that would prevent binding-induced breakage. So there are kind of like two things you can do to inhibit binding-induced breakage here. Yeah, but definitely that's like the central actionable thing that you can do to capitalize on this mechanism. Yes.
Thank you, Dr. Lee.
All right, this question is from Alexandra Levine, who is joining us virtually. Are there, and this is a question for the panel, I think Dr. Cohen can start us to answer this question. Are there any data yet regarding co-infection with EBV and COVID in terms of possible issues with long COVID? For example, COVID and reactivation of EBV or vice versa?
Yeah, so I seem to recall that the literature shows that some individuals with long COVID tend to have higher levels of EBV DNA in their peripheral blood than otherwise you'd expect. And I think at the present time, it's not, I sort of assume this is reactivation, but it could be much more complicated than that, much more complex. But I think there is an association with, again, elevated EBV DNA PCR in the blood of some of these individuals.
Thank you. So I have one question for Dr.
Sure.
Lee. So I'm Jewish.
Okay.
So you know. So Jews have been around a long time, and there are Eastern European Jews, and there are ones, they're called Sephardic Jews.
Okay.
So you mentioned about the difference between I think Asians and Jews.
Yes, it's Chinese.
But there may be that if you looked,
Mm-hmm.
there may be differences between those who are more come from Southern Europe and those that come from Northern Europe.
Agreed. Yeah. So the two Jewish genomes that we looked at were Ashkenazi Jews. And I'm not too familiar with, yeah. So definitely we've only looked at 4 genomes between just 2 very, representative ethnicities. So there's a lot of work to be done there. And with long read sequencing that now allows us to actually look at these tandemly repeated sites in our genome, it's a really exciting time for this discovery to come out as well. And recently there's the pan-genome paper that was published, which consists of 47 long read sequence genomes. That's representative of most of the ethnicities in the human population in the world. And so that's a great starting point for us to really dig into how polymorphic this site is and what is the range of copy numbers that we're looking at, and then what does that mean in terms of susceptibility to breakage. So those are all things that definitely we want to investigate. Yeah.
There are a few things left, huh?
Right.
Well, thank you again. And I imagine we're at the lunch hour.